cover in A&P 1. And this one, I think, is kind of interesting. At least students seem to find it interesting. This is all about the special senses. So we didn't talk a lot about the touch senses, just because there's a lot of information in that sense, and there's multiple receptor types that all work differently. So you may have noticed that I kind of glossed over that, even though your book paid it more attention. But the special senses, we do spend a little bit of time talking about and seeing how they all work. So that's olfaction, that's the sense of smell, gustation, sense of taste, and then hearing and equilibrium, which both take place in the inner ear. And both, what's very interesting is they use very similar methods, even though they do very different things. So the way we detect sound and the way we detect motion actually happen in the same way, just in in the same organ, essentially, as we'll see later on. And then vision we'll probably talk mostly about next time. All right. So in the special senses, we have two general wiring patterns, so to speak. In all of the senses except olfaction, so I just said the non-olfactory neurons. So this is vision, hearing, taste, all the rest. We have this pattern, where we have a specialized receptor cell, and that receptor cell, its membrane potential changes according to the stimuli that are present. But what you'll notice is that this receptor cell itself doesn't have any connection to the nervous system. So instead, for all the non-olfactory neurons, we have this two-cell setup, where we have a specialized receptor, and then we have a transmitter cell, so to speak. And this cell detects the membrane potential change here, and then creates a signal that is sent into the central nervous system. So if we look at the membrane potential of the receptor cell, when there's a stimulus, we see a depolarization, right? Not an action potential, like we've seen with neurons, but we do see a change in membrane potential. That change is detected by this neuron here, and so here's the stimulus. There's a little bit of a delay so that while it's detecting, but then it starts sending a signal that something has occurred at this receptor cell. Now, in the olfactory sense, we don't have these specialized receptors. Instead, the neurons themselves do the work of detection. So a stimulus comes in and directly stimulates the dendrites of this olfactory neuron, and it starts sending a signal as soon as the stimulus is present. So we'll see here in just a minute that the olfactory neurons are kind of unique in the body because they're always reproducing. We all learn that neurons don't divide. Well, the olfactory neurons are a rare exception to that. But it's the nerve itself that is doing the detecting. For everything else, we have a specialized receptor cell that's doing that detection work. All right. So we'll start with the olfactory tract. So here's the nasal cavity, right? Here are the nasal concha, which you'll learn more about in the respiratory chapter. These are basically curvatures on the inside of the nose that expand the surface area so that as air comes in and goes down into the lungs, it gets humidified and warmed. So one of the reasons we have a nose in the first place is not just to smell things, but it's to humidify, filter, and warm the air that we breathe. So by the time the air gets to the lungs, it's about body temperature and 100% humidity. But at the top of this nasal cavity, we have the olfactory organ, which is made up of the olfactory epithelium and then the transmitter cells that go along with that. All right, so the sensory neurons that make up the organ of olfaction are found here. Their axons kind of bundle together into about 20 bundles that pass through this bone called the cribriform plate. When you were learning about the skull, you should have seen that. When you look down in the front part of the bottom of the cranial cavity, there's a whole bunch of little holes in the middle. Well, what passes through those holes are the axons of the olfactory epithelium. So that's here. They go up there. They then synapse in the olfactory bulb. The olfactory bulb, remember, is cranial nerve 1, right? It's found at the base of the frontal lobe. So here's the frontal lobe. And right along the bottom is this olfactory bulb, kind of an expansion at the end. From there, the information goes through the olfactory tract, which is right here. And then it projects to multiple places in the brain. Now, so olfactory information goes not just to the olfactory cortex. That's where we experience smells. So that's our conscious understanding. It smells like a rose. It smells like strawberries. That comes from the olfactory cortex in the cerebrum. But then, in addition, the olfactory tract projects to the hypothalamus, and it also projects into the limbic system. And it has particular associations like with the hippocampus, which is where memory is made. So smell, unlike the other senses, can have a profound impact at multiple levels of the brain. A smell might trigger a memory, a smell might trigger an emotion, even if you don't know why. And the reason for that is because of these separate connections to different parts of the brain. Now, why on earth would that have evolved? Well, in the animal kingdom, we see smell, odor, being used for all different kinds of communication, advertising, territory marking, all those different kinds of things. So it's this cross-wiring, so to speak, is probably how that got to exist, that animals use smell to communicate at multiple levels. Now, the interesting thing is some research is finding out that those same kind of things may affect us too. We have science has sort of said in the past that humans don't have pheromones or scent communication molecules, but it's looking like we actually do, in fact, have pheromones. We just don't experience them consciously, but we experience them unconsciously. So new studies going on about that. All right. If we zoom into that olfactory epithelium, it looks a little bit like this. So here are those bundles through the cribriform plate here.
So it's upside down on this thing?
No, the skull is here.
Oh, okay.
And the epithelium is down here. So, it's still right side up. So the ends of these olfactory neurons, you can see one here. So here's the cell body of it. Here are the dendrites, right, that are kind of spread out along the surface. So this is the inner surface of the top of the nose, is right down here. And then here you can see a cell that's still developing. Because I remember I said that this is one of the few places in the body where neurons divide and differentiate so that the olfactory neurons are constantly being replaced. Now, why do they need to be replaced? Because they're exposed to the outside world here. The air that's coming in from here has direct access to these dendrites. Anytime you have that situation, cells have a limited lifespan. The outside world is damaging to our cells. So in order to keep the olfactory neurons fresh and working properly, they have to be replaced all the time. Just like you'll see like in the GI tract next semester, you'll see that pattern is present there too. All right, so these are the sensory cells. They actually are going to detect molecules in the air. So, how does the sense of smell work? There are molecules in the air that are detected by these olfactory neurons that then send a signal up into the brain. Now, sharing the space with the neurons, though, are these epithelial cells. So here's one, here's another, here's another. These are standard respiratory epithelial cells, so pseudostratified, columnar, and then they're ciliated. So the neurons kind of are squeezed in between there. All right, covered everything. Oh, the dendrites are not hanging out there in the air. They're actually suspended in a mucus layer. So this yellow stuff here is mucus. It's produced by this gland right here called the Bowman's gland. So in order to detect molecules in the air, those molecules first have to dissolve in this mucus. So the things that we smell most easily are things that are water soluble, because those molecules dissolve in this mucus and then can be detected by the olfactory neurons. All right, so that's olfaction. Gustation, in many ways, is very similar to olfaction because they do the same thing. Olfaction is about detecting molecules in air, but of course those molecules in the air have to dissolve in solution in that mucus first to be detected. Well, gustation is pretty much the same thing. It's detecting molecules in solution. So even the brain, the gustation and olfaction senses are very closely linked. When you have a stuffy nose or a cold, things don't taste right or things don't taste very good. It's because taste and smell sort of work together in the brain to bring information about food in particular. All right. So we have 3 different types of taste buds. The most sensitive are these circumvallate papillae, which are found here at the back of the tongue. They make a kind of V. And we'll talk more about those in a minute. But there are these deep epithelial folds, and then the taste buds themselves are here off to the side. And we'll zoom in on this one in a minute. The fungiform just means mushroom-like. You can imagine that looks a little bit like a mushroom. Well, those are found sort of in this area of the tongue in the middle. They have about 5 taste buds each, so they're not as sensitive as those ones in the back. And then the filiform papillae, which are found at the front of the tongue, they are sort of hair-like or feather-like, and they actually don't have taste buds in them. Instead, they just provide friction that helps the tongue to move things around, like while chewing and swallowing and things like that. Now, the different, you can take all tastes and divide it into 4 basic categories, and the tongue senses these 4 things separately. So there's sour and bitter, salty and sweet, which I'm sure you've heard that before in previous science classes or health classes. So those are the 4 that we hear about all the time. There are 2 more, though. We sense water. So water is essentially another taste type because, obviously we need water to live, and we need to be able to assess the quality and clarity and healthiness of the water that we're drinking. So water creates its own sensation of basically water. And then the most recently described of the taste types is this thing called umami. Umami is a meat sense. It's stimulated heavily by things like, chicken broth, beef broth, meat. So it's that taste of meat is essentially your umami sense. All right. So if we zoom in on one of those circumvallate papillae, so we have these deep epithelial folds here, and then we have this bundle of nerves here in the middle. Now, if we zoom in on one of these guys, these are actually taste buds. And they're called taste buds because if you think of this nerve as kind of a tree, these little extensions look like buds on that tree. So that's how they get their name. Taste buds is because they look like buds when you look at them under the microscope. So if we zoom in, we see a structure like this. So this is the taste bud, all right? Now on the side, on the epithelial side, so food, dissolved molecules are going to come and work their way down this fold. And those molecules get detected here at the taste pore. So the taste bud is kind of, bud-shaped, and then at the end we have this pore. In that pore, this opening, we have the taste hairs. And this is where the actual detection occurs. Anytime you have hair-like projections, it's a surface area expansion. The cell, there's a lot more cell membrane here because of this hair-shaped end than there would be if it was flat. So it gives a big surface area for the detection of molecules in solution. All right, so this is the receptor cell. So we call them gustatory cells. They have hairs at one end, and then they're long and skinny. And then at the other end, here we have the axons of neurons. So remember in that first slide, all the cells but the olfactory cells have receptor cells and then transmitter cells, right? So these are the receptor cells. These are the transmitter cells out here. Now, the gustatory cells are being replaced all the time. So we have at the deep part of the taste port these things called basal cells. They divide and become new gustatory cells, so new taste cells. And then interspersed with the taste cells, the gustatory cells, are these things called transitional cells that basically support the gustatory cells. So taste cells, gustatory cells live several months. As we get older, we have fewer and fewer of them. So if any of you have been around young children, we often find them to be rather picky in terms of what they eat. One of the reasons for that is they taste more. Because they have many more taste buds than the adult, things that we think are mild are actually, very bold taste to the child. So what you often see with kids is that left to their own devices, they choose a kind of bland diet, and it's simply because they taste more than the adult does. It's another reason why, what you like to eat now and what you like to eat when you were 5 may be quite different. The reason for that is you taste differently because you have fewer taste buds as you get older. This also comes up in the elderly. Sometimes the elderly don't take in enough calories because nothing tastes good. And that's, again, because the gustatory cells have not been replaced as vigorously as when they were younger. All right. So our first question of the day. All right. So hopefully this will work. This part will work. All right, taste buds. What is true about taste buds? Not the greatest of questions, I'll agree on. All right, so the best answer there is C, can only perceive taste if the molecules are in solution. So one of the reasons that our bodies produce saliva, that the liquid that enters the mouth, is to accomplish this, to dissolve the food that we're eating in solution so that it can be tasted. If you've ever had a case of super dry mouth, taste doesn't work right unless there's enough solution there to dissolve those molecules. So it isn't 7 basic tastes, it's really 4 or 5 or even 6 if you include umami and water, but not 7. They last longer than 30 days. It's every couple of months they're replaced. And obviously they're only found on the superior part of the tongue. And taste buds don't generate their own action potentials. They need those neurons to generate action potentials into the brain for them. All right. Which of the special senses contains receptor neurons that are the only nerve cells in direct contact with the outside environment? Take it that's everybody. All right, correct answer is C, smell, olfaction. Only neurons to be exposed directly to the outside environment. Because while the inside of the nose is still inside the body, What's inside the nose is air that's come from the environment, so it's unaltered. So it's the only place you see that, and you see those dendrites kind of in that mucus waiting to detect odor molecules. All right. So taste and smell, a lot of similarities, detection of molecules in solution in essentially two different ways. All right. So equilibrium and hearing. Another two senses, obviously these two are very different, but they occur in much the same way, just like olfaction and gustation do. So both equilibrium and hearing depend on the actions of these specialized receptor cells called hair cells. So the hair cells get their name because on their top surface they have hairs or cilia. They have a bunch of stereocilia that are get increasingly taller as you go to one side of the cell. And then there's one long one that has some additional stuff inside, some microtubules, called the kinocilium. So the motion of these hairs, if the hairs bend this way, that will cause inhibition of this hair cell. So its cell membrane potential will go down. If the hair cells bend this way, the hair cell's membrane potential will go up, so it'll depolarize. These are fascinating, structures because essentially they're mechanically gated sodium channels. So when they bend in one way, more sodium enters the cell, but when they bend in the other, less sodium enters the cell because it actually pushes those channels closed. So the movement of these hairs are how equilibrium and hearing work. And the way that this is accomplished is by getting this fluid that the hair cells are suspended in to move one direction or another based on the anatomy of the thing. All right, so then the hair cell, which is not a neuron, but the change of its membrane potential in response to the movement of these hairs is conveyed to the CNS by this sensory neuron here. So this neuron wraps around the hair cell, and when it changes, the sensory neuron changes and starts sending action potentials into the brain so that the whatever has been detected out here can be brought into the brain and processed. All right. All right, so a little bit of ear anatomy. We divide the ear into three regions. The external ear is the part that you can see from the outside. The middle ear sits between the tympanic membrane and the oval window, which we'll talk about in a minute. And then the inner ear, this is where the actual sensation of hearing and equilibrium is developed. So the inner ear is this kind of elaborate structure of bony pathways, tubes inside the temporal bone that contain the organs of sensation. All right, so if we start at the external ear, the part of the ear that we can see most easily is called the auricle, also called the pinna, so that's the ear part that you can see. It's made up of elastic cartilage, so it flexes and bends very easily, but it snaps back to its original shape. You can bend your ear and it'll go right back to its normal shape. The shape of the auricle is not by accident. Sometimes we think that ears are sort of funny looking. How on earth did they get to be that way? Well, they're actually tuned to the frequencies and pitches of sound that we use in communication. So one of the reasons, like in a crowded restaurant, you can fairly easily hear the person you're talking to is because of the shape of the auricle. It has a tendency to funnel the frequencies that we use to communicate with each other, and it helps to keep out the frequencies that we don't use. So that funny shape is not by accident. The external acoustic meatus is the canal that connects the outside to the middle ear, and then it ends at the tympanic membrane. And the tympanic membrane is a thin, taut, relatively inelastic, it usually is white or kind of pearly gray membrane that completely blocks the ear canal. And it acts as a kind of diaphragm. Sound comes in here. Sound is vibration, right? and it causes that tympanic membrane to vibrate, just like the head of a drum vibrates, or a speaker vibrates. And then that vibration at the tympanic membrane is conveyed to the cochlea, which is the hearing organ, by the auditory ossicles, which we're going to zoom in and look at that more in a minute. So we get to the middle ear. The middle ear is a sort of interesting space in the body because it's not connected to the outside, but it's not exactly inside the body either. So because the tympanic membrane is sort of blocking this path, there needs to be a way for stuff to get from the middle ear out. You know, this is all mucous membrane here, so there has to be an exit. So that exit is what we call the auditory tube. It used to be called the Gustation tube, and it connects the middle ear here with the back of the nasopharynx. Now, when everything is healthy, every time you swallow, this tube opens and closes to allow air pressure to equalize, you know, between the inside and the outside. When this auditory tube opens, air can rush in or go out, depending on the relative pressures. And also, any fluid that might be in the middle ear can exit through the auditory tube when we swallow too, because that tube opens and closes. When you have a cold, however, this tube can end up clogged and can create inner ear, middle ear pain. This is the space that fills up in ear infections commonly too, called otitis media, or a middle ear infection. But that auditory tube is supposed to keep the air pressure in here, same as out here, and keep this empty. All right. So let's zoom in and look at that middle ear. All right, so tympanic membrane here. It isn't perfectly flat. It actually has kind of a dish shape to it. It's a little bit convex towards the middle ear. And then we have the three tiniest bones in the body, the malleus, the incus, and the stapes. Now these three little bones connect the tympanic membrane with the oval window. All right, so what's an oval window? The cochlea, the organ of hearing, has two windows. One that allows sound energy in, and the other that allows sound energy out. So the oval window is the input to the cochlea so that we can hear. So as the tympanic membrane vibrates, the malleus vibrates, the incus vibrates, and the stapes vibrates, and that vibration causes the oval window to vibrate. All right. So why make it so complicated? You know, why did we evolve with this transmission system instead of just having the oval window be in direct contact with the outside world? Well, one of the reasons is because of these muscles right here. So the tensor tympani attaches to the malleus, and the stapedius attaches to the stapes. These muscles allow the ear itself to control how much sound energy is passing through it. All right? So the tensor tympani, for example, the tighter the tympanic membrane, the harder it is to move it, right? You know, it's like a drum. If you tighten the head of a drum, the sound is not as loud, but it's higher pitched. Well, the tensor tympani does that with the tympanic membrane. By tightening it up, it makes it harder to move and essentially turns the volume down. If the tympanic membrane is harder to move, less sound energy will be conveyed from here to here. So that's one approach, tense up the tympanic membrane. Another is at the stapes. The stapedius pulls the stapes away from the oval window. So let's say you're in an environment with very loud sounds. You know, you're standing right next to a big waterfall, for example. The tensor tympani will tense up the tympanic membrane. The stapedius will pull the stapes from the oval window, and essentially the loudness will go down because there's less sound energy actually making it from here to here. Another more common example is if you've been to a loud club or a rock concert, you might notice that your ears, you hear differently for several hours afterwards. That's the stapedius and the tensor tympani. They have turned the volume down So then when you go to a quiet room, everything sounds funny. So these muscles are rather slow to respond, so it can take several hours or even several days for the system to get back to its normal volume setup. All right, so that's why the complex transmission structure there. Okay, so then we get to the inner ear, and the inner ear has a very complex shape, and it should, because it's the shape that makes it work. All right, so we can divide this complicated inner ear into two big pieces. This snail-shaped piece over here, this is the cochlea. So from about here on over, that's all cochlea. And then we have the semicircular canals. And you have to remember, this is a three-dimensional structure that's being represented in 2 dimensions on the screen. Okay? So we have one semicircular canal, and here's another. And then this one actually would be coming out at you. Okay? So we have an X, a Y, and then a Z plane that would be coming out of the board at you. And all of those semicircular canals join together at what is called the vestibule. So this region right here is the vestibule, and that's where the sensation is actually created from we'll talk about that in a minute. All right. So all of this complexity, this complex shape, is all contained in bone. It's all in the petrosal part of the temporal bone. So just above the mastoid process is where you're going to find this elaborate network of channels. Now, what do we find in these channels? A substance called endolymph. It's one of the fluids of the body that you hardly ever hear anything about, except when you're talking about the ear. So we have blood, we have cerebrospinal fluid, and we have endolymph. So endolymph is a little bit like plasma. In other words, it has lots of sodium, not very much potassium, but it's a little more viscous. In other words, it's a little thicker than the very watery plasma is. But the endolymph that fills these channels is shared. You can see that the cochlea is attached to the vestibule here. So this attachment means that this endolymph can move freely from one region to the next. All right. So what we're looking at here, this is the bony labyrinth. A labyrinth is a maze. That's what that word means. And you can imagine early anatomists looking at this and seeing its complexity would say it's maze-like. So the bony labyrinth is the bone, and the tracts that have the endolymph, we call that the membranous labyrinth. So inside the bony labyrinth is the membranous labyrinth. And it's inside the membranous labyrinth that then the sensations of equilibrium and hearing are actually made. All right. So if we take the bone away, this is what we have. All we have left is the membranous labyrinth. So we can divide it into the cochlear duct. The cochlear duct is a spiral. It starts and ends at it starts at the oval window, and then it ends at the round window, and it kind of winds up and then winds down, as we'll see here in a minute. Cochlea means snail, and the cochlear apparatus really does look like a snail. So that's one whole side of it. And then the other side of it is the vestibular complex. So this is where equilibrium comes from. We have our 3 semicircular canals in 90 degrees to each other. And then we have the utricle and the saccule, which are found here and here. And the semicircular canals give us information about rotation. So when you shake your head no, for example, you can feel that, and that's because of movement in the semicircular canals. The utricle and saccule give us information about acceleration and gravity. And I'll show you some examples here in a minute, but just to orient you. Okay. And all of these ducts contain that endolymph. All right. So first up is the semicircular canals. Okay, how do they work? Well, in the ampulla of the semicircular canals, so here's one, here's another, and here's a third, we have this structure called in the vestibule, we have this ampulla. So if we zoom into one of these ampulla, what we see is we have this kind of gelatin wedge called a cupola that almost, but not completely, comes all the way up to the edge of that tube, of that semicircular canal. So the top of this cupola can move. So it can flex this way or that way. It can bend this way or bend that way. And embedded in this cupola gelatin are hairs of hair cells. Remember, hair cells change their response according to whether hairs bend one way or bend another, right? So when this cupola moves this way or this way, the hair cells will detect that movement, and then the sensory neurons that are connected to them will then transmit that information to the brain as a change in motion. So as we move our bodies around, the fluid in these semicircular canals gets to moving. It moves relative to the body itself. So when the fluid moves this way, the cupola is pushed this way, or vice versa. All right, so here's what we mean by that. Okay, let's look at the head. Here's the semicircular canals, and we're going to take the one that's in that's parallel to the ground, right? Okay, so that's the one we're looking at. When you rotate your head one direction or another, the endolymph doesn't move as fast as the bone that it's contained in. If you take a glass of water, for example, and you spin it, you turn it like this, what you'll see is that the water never moves as fast as the glass does, right? Because water is fluid. It can move separately from the glass. We see that same effect in the semicircular canals. The endolymph moves less quickly than its surroundings, so we get a relative motion. We get fluid moving this way or fluid moving that way. At the ampulla, the cupola is bent one way or another, and that bending sends the signal to the brain that the head is rotating in one direction or rotating in another. All right. And we have 3 semicircular canals, and they're approximately 90 degrees to each other. So the way your book describes it, I think, is pretty good. One of them senses the yes direction. So when you nod your head yes, that's a rotation in this plane, right? One is in the no plane, so that's this one, right? So yes is this one, no is this one. And then the last one is for head tilt. When you tilt your head to one side, that's a 3rd semicircular canal plane. So all of these things working together give the brain not only information about what the head is doing, but also information about what the body is doing. So when we were little, we all played on merry-go-rounds, right? So once you get going on the merry-go-round, the endolymph in your semicircular canals gets to moving with the merry-go-round, right? So then you get off the merry-go-round, your body has stopped rotating, but the endolymph is still rotating, right? So you have that sensation of dizziness or rotation, and it all comes from these semicircular canals. So it isn't just head position, it's body position too. So in things like weightlessness in astronauts or in airplane pilots and that kind of thing, this rotational information becomes hugely important. As it does in things like gymnastics or tree climbing or skiing or any of those things. So rotational movements. All right. The second part of equilibrium has to do with gravity and acceleration. We need to know not only in which direction is our body spinning, so to speak, but we need to know, is it slowing down, is it speeding up, and what's its orientation to gravity. If you bend too far over, you fall down, right? Well, you need to know that before that happens so that you don't have any kind of injury. So that information comes from the utricle and the saccule. So here's the vestibule, or the vestibule, and here are those ampullas of the semicircular canals that we talked about. So in between those two, we have these different organs. So the utricle is here, and the saccule is here. Now here's our hair cells again, right? The hair cells, and they have their hairs embedded again in this kind of gelatinous material. But instead of being, cup-shaped like the cupola, on top of this gelatin are these little tiny rocks of bone called otoliths. Oto is ear, lith is rock. So this is quite literally ear rocks. These are crystals of calcium, some of the same stuff that bone is made of. And they're sort of embedded, these otoliths are embedded in this gelatin here. Now, the otoliths have some mass to them. They're very dense. So, mass is what's affected by acceleration and gravity, right? So, and this is gelatinous. So these otoliths can pull the hair cells this way, so they'll bend this way, or they can pull the hair cells the other way. I think I've got it. Yeah, here we go. All right, so when the head is stationary, the otoliths are pulling, or the gravity is pulling down on the otoliths, and there's no motion at the hair cells, right? But let's say you tilt the head back. Now the otoliths kind of slide, but the gelatin is firm enough where they don't slide away. They just sort of slide down, and they pull the hair cells with them. So this same thing occurs in acceleration. So you're sitting at a red light, it finally turns green. When you step on the gas, your otoliths lag behind the rest of the head, and the brain detects that as acceleration. Same with deceleration. You slam on the brakes. The otoliths keep going for a little while and pull those hair cells then in the other direction. So the otoliths at the utricle and the saccule bring us information about our orientation to gravity. In other words, are we upside down or right side up, so to speak, and linear acceleration. So that speeding up or slowing down that comes with the this relative motion. All right. So like when you ride a roller coaster and you go down a great big hill real fast, you feel that sense of weightlessness. It's because your otoliths are now actually being pulled up off of the underlying hair cells, and that's detected as falling. So it's that sensation of falling. All right, so the vestibular system. So semicircular canals, and then the utricle and saccule for linear acceleration and gravity. All right, so all that's left then for the inner ear is hearing. Well, that shouldn't be too hard, right? Well, hearing is probably one of the more complex of the senses in terms of how it works. But your book does a pretty good job with presenting kind of the basics. All right. So here are the semicircular canals. We're done talking about them. The other part of the inner ear is the cochlea. And it's a spiral that kind of winds up to a point and then winds back down. So if we take the cochlea and we cut it up and down, we see a pattern that looks a little bit like this. So, a snail shell, right? The snail is in a tube in that shell, right? And that tube winds around to a point. Same general pattern for the cochlea, only there's room for two snails in the cochlea. So it's actually two tubes that wind together up to a point. And the way that sound energy travels in here is, you have blue arrows and orange arrows. So sound comes in, blue arrow or purple arrow, and it travels up to the top, and then it travels back out. So it spirals in, and then it spirals out, and then it goes to the round window. So we go from oval window in to up to the top, and then from the top down and out the round window. Better pictures coming in a minute. All right. So we have three ducts. So room for two snails, right? One of those is the vestibular duct. So the vestibular duct is the input duct, and the tympanic duct is the output duct. So we have sound information comes in and travels up the vestibular duct to the top, and then it spirals back out through the tympanic duct and out the round window. In between those two, we have another duct called the cochlear duct. This is where the endolymph is. Well, actually, all of these ducts are filled with endolymph. I'm sorry. This is where the organ of hearing is, the organ of Corti, which I'll show you in a minute. All right. So the tympanic membrane moves. That moves the stapes. The stapes moves the oval window. Sound energy enters. It travels up the spiral and then back down the spiral. And then sound energy leaves the inner ear through that round window. All right. And it's the relative movement of the endolymph in these cochlear ducts that actually produce the sensation of hearing, which I'll show you in a minute, if I know we're about out of time. All right, actually, I think we'll stop right there, and then we'll do the whole hearing thing next time.
So what is a cochlear or a cochlear implant do? Because I know a lot of hearing impaired kids in elementary school, and they would always have both. So I know that there's an implant, there's a magnet that's on the inside of the skull, and there's one on the outside of the skull.
Right.
So what exactly?
Basically, a cochlear implant, it bypasses the cochlea.
Okay.
So it takes sound energy from the room and translates it into nerve energy that goes into the auditory cortex. So it takes the place of the cochlea.
Okay.
Because the cochlea turns sound energy into CNS impulses. The cochlear implant does the same thing.
Okay.
It's one of the reasons why they work best if they're put in early. Because the child has to learn what that information means, because it isn't perfect. It isn't the same. But the brain can learn to associate that cochlear implant signal with actual hearing.
So in hearing impaired people, is that what's wrong? Is their cochlea is just not?
Yeah, and most congenital hearing, congenital deafness, it's because of a cochlear problem.
Okay.
So replace the cochlea and you could give hearing back. Now, nobody thinks it's as good. It's adequate. These kids learn to talk and they learn, they can play instruments and things like that. But nobody thinks it's quite as good as what the cochlea does naturally. But yeah, it's cool because there's an exterior and an interior piece to it. The exterior piece has the battery too. So it's inductive. The electricity on the inside comes from the outside through the skin.
I just always remember because they would fall off and then they just put it right back on.
Yeah, it has a magnet in it. Yeah. Yes.
Friday. I'm going to be out of town, so everything's going to be on integrity, right?
May 10th.
It's like
That'll be the final exam review.